So these are the folks that are here. I wanted to sort of talk a little bit about what we're trying to discuss today. So we've heard a bit about neuroinflammation. It was introduced in the last topic. I think one of the themes that we haven't spent much time on over the last sort of morning is to focus in on the neuropsychiatric symptoms. So think about some of the components. We heard a little bit from the insights in the discussion in the excellent patient forum about some of the differences and how these are very unique. But we haven't really spent any sort of quality discussion thinking about where these intersect. So where do these symptoms come in, and where do they play in the various roles, and how do they link in terms of early symptomatology? So what I thought I would do to start off with is to pose a question out there, and maybe I can start off with you, Kerry, asking about, so tell us a bit about, obviously your depth is significantly in the psychiatry and depression side, but tell us a bit about what we know about depression and how it can link in to obviously something as broad as neuroinflammation, but then also potentially how we can link it into Alzheimer's disease.
Sure. So when we think about depression in the current nosology, it's really the realm of symptoms related to sadness, anhedonia, suicidality, vegetative symptoms, really lack of hope. But over the last decade or so, there's been an enormous amount of progress, both in the basic neural circuitry of striatal-based processes, as well as amygdala and prefrontal processes that relate to the stress response, the anhedonic response. And these are also very much associated with the hippocampal roles of contextual memory formation, episodic memory formation. And tied within all of this is a large amount of evidence now in dysregulation of the inflammatory system, with peripheral biomarkers of CRP, IL-6, TNF, all being very increased in depression states, and increasing data that a past history of stress, trauma, and depression significantly raise risk for Alzheimer's disease and dementia. And so I think one of the things that we'll want to talk about today is how can understanding these disorders of stress and depression lead to a better understanding of additional risk for dementia.
Anything you want to add to thinking about sort of from your perspective as well?
No, I mean, I think opening up neuroinflammation, neuroimmunology, and depression has opened up a whole new area. As you know, the field, a lot of the companies have moved out the psychiatry field, moving away from the monoamine hypothesis and failures in novel hypotheses, but ultimately failures such as targeting neuropeptides, CSF1 and so on, CRF1. So for us, opening up, there appears to be a really compelling story, I think, for the role of inflammation and in depression, at least a subgroup of patients. And we've certainly been working along that line, working with a number of people in this room on the various consortia in the UK to try and identify, you know, what are the markers, what are the subtypes, and what are the right molecules to test.
Thinking about the angle of, I mean, you brought up the angle of funding here, because we've heard a lot today about the needed funds for how you can think about large diseases like Alzheimer's disease. How do we take advantage from a perspective of focusing in on the neuropsychiatric components, what's needed in order to be able to get us to a place where I think we can take you know, sort of real learnings from other areas of the field and be able to bring that to an area that still is very much part of the symptoms, but isn't necessarily yet at the forefront.
Right. Well, I mean, I think the current separation between psychiatry and neurology is, you know, an artifact of hundreds of years of history that we don't need to go into, but I think where we currently are now, and was really alluded to at the end of the last session, was the need for better behavioral biomarkers. And so I think one way to do that is looking for where we can leverage opportunities, either bringing psychiatric expertise to have better behavioral and digital phenotype biomarkers in the dementia space to understand those intersections, as well as opportunities for repurposing compounds, be they targeting neuroinflammation in MS or Crohn's disease or potentially Alzheimer's disease, to look for places where you might be able to open up additional markets that are really affecting, you know, billions of people in the depression anxiety space.
And so on to that point then, so I mean, I guess that's where we are, certainly in Janssen. How can we, the thought about neuroinflammation and depression is not new. I think what is new is the advances in the tools available to us. As immunology have identified novel drugs to target very specific aspects of the innate immune system, then potentially we can take those and either use them as tools or repurpose them for treatment. I mean, you asked the point about funding. I mean, one of the things that we've managed to do is work very closely with funders such as the Wellcome Trust and MRC about how you can actually work as a consortium. So, for example, we're working on a Wellcome Trust consortium called Neuroinflammation in Mood and Alzheimer's, NEMA. And this is a fantastic, I think it's a model of how to do this. It's 4 different pharma and 7 different UK academic groups. And the idea is we're really pitching in our, and as you know, your group's involved in that, we're really pitching in expertise, shared expertise, assets and so on, all with the aim that we can go back to the Wellcome Trust and say, hopefully we can identify the right subjects and the right molecule and then move forward and test that drug in the right population. The MRC, again, we got a great example, I think, of a consortium where for very little money, we were able to bring a group of people together and really sweat the old data, really go and reanalyze existing data sets. So there was some de novo data generated, but mostly it was go back to the data sources, go back to our immunology trials from both GSK and from Janssen, and really understand where the molecules that had effects upon mood. And for very little cost, I think we've actually generated a significant advance for the companies and for the academics.
So let me just follow on that theme as well, and maybe I can also ask sort of in the audience what sort of representation we have of those that are part of the funding bodies. So do we have anybody who actually is involved in funding that can sort of bridge some of the discussions? So, well, there was one at least. So that's good.
Yeah.
We lost them to the other panel.
I guess so. All the money went out the door. That's good to know as well. So, I mean, along those lines, I mean, obviously, clearly, in order to build these types of consortia, there still needs to be a really sort of clear understanding of what the endpoint is. So, what it is that you're trying to achieve. You know, the idea of repurposing I think is great, but ultimately you need to, and we heard a lot about sort of polypharmacology this morning as well. I don't think anybody really believes that ultimately you will be delivering a single therapeutic that will treat all of the symptoms of many of these diseases. And the other piece I think we heard loud and clear is that we've known a lot about the amyloid hypothesis. We're now, I think, very much in the conclusion that in order to be able to be successful, you're going to have to be very much earlier in the disease as well. And let me ask the question around, do we feel that is the same for the psychiatry components? I think we talked a bit about certainly where psychiatry appears once a disease has been diagnosed, but what about risk factors? So that's clearly something that has been discussed from the standpoint of multiple sort of inputs, but what about specifically from psychiatry? And do we feel that there's a really sort of tangible sort of direction to go from a risk factor standpoint as well?
I think it's very much being collected. I think there's several lines that are intersecting here. One is history of stress and trauma, starting with childhood trauma, but really whether there be traumatic brain injury to other ranges of trauma and stress or risk factors for everything. And I think in addition to risk factors for dementia, we're starting to understand a great deal about their role in risk factors in the neural circuitry of depression and stress disorders. And the other aspect of that is biomarkers. You know, we talk about depression, stress, anxiety. You know, there's only a handful of historical psychiatric clusters, and yet we know that these are really many, many, many diseases. We talked about last night at dinner, you know, there's probably 10 different diseases underlying amyloid plaques, and there's probably 100 underlying the depression symptomatology. And we've got to be able to get at precision medicine with biomarkers that will tell us what is an inflammatory depression versus what is not. And then amongst the inflammatory depressions, what are the subtypes there? And just one thing I want to throw out there, and it was mentioned in one of the earlier groups about being in the pre-competitive space for collaborations. And I want people to be aware of the Psychiatric Genomic Consortium, which has brought together really many tens of thousands. And there was just a paper out with the largest GWAS for depression with over 100,000 total with about 25,000 cases. And the PTSD space, we also have, are aiming towards 75,000 at the end of this year. So there are these large multinational collaborations that have samples that have deeply phenotyped cases that I don't know of anyone who's really tried to cross the dementia space into either the depression or PTSD large collaborative spaces, and that would be one place to look for large data sets in the pre-competitive space.
So how do you think we do that? I mean, what is this a case of getting the right people in the room at the right time, or is it a case of actually showing specific targets that clearly ultimately impact disease progression? I mean, you know, there's obviously these camps have existed for a while. I think there's been a lot more of bringing together of certainly the understandings, but I still feel that we haven't quite sort of figured out how to match So any thoughts about how we can do that?
So I think it is happening. I was struck this morning by David Mayhew talking about there's probably 4 oncology scientists for every one in neuroscience. Well, probably about 90% of those are working in anything but psychiatry at the moment. So I think we do need to rebalance. We need to really look at mental health psychiatry research and rebalance some of this. And I think there are methods that we can use from what drove Alzheimer's and dementia research. So in the UK, we have the Dementias Platform UK, which provides a data portal, an infrastructure for that, an imaging infrastructure and stem cell infrastructure. Now, there is probably room of doing something very, very similar to that, particularly for the data. How can we actually, again, sweat the data that already exists rather than just generating new data each time? How can we put data sets together in a way that is easy for academic researchers and industry researchers to access? So I think there's room for that. I think there are examples where this is going very well. I talked about NEMA in the UK. There is a biomarker initiative in the US, FNIH, which again is working with a lot of the same people, working with the NEMA consortium. And the idea is, can you do this in parallel and then eventually, hopefully, join them together? I think there is a groundswell to do this. It's actually just getting the people together and actually making the case to the funding bodies.
So before I move into the next theme, I'm going to put the audience to work. So questions. Now, we don't have the system. You don't need to log in. You don't have to type anything.
Just yell.
It's quite old school. All you have to do is raise your hand, shout out. You could actually even come sit on the stage. But either way, so I thought I would, from what you've just heard, obviously we're looking at these intersections. Questions from the audience that we can bring into the discussion as well.
Maybe a really naive one, but what is the case that we're talking about? Symptomatic treatment or
I think it's all of the above. I mean, I think there's a full set of pipelines that are not being currently, that are a gap in the field. Everything from really, there is currently a very good basic science understanding of circuits that we can approach with all sorts of new circuit molecular biology, all the way up to repurposing already FDA-approved compounds and the things in between. I think one specific thing that we've been talking about here is targeting the large-scale data sets that show increased inflammation in certain subsets of depression. And there hasn't yet been a multi-site trial, even with, there was one with infliximab, but they didn't do precision medicine. There's a number of other potential good anti-therapeutics targeting TNF, CRP, IL-6, and others, for which there's a lot of preclinical data, for which you could say, let's take a precision medicine subtype of people with severe depression, even severe suicidal depression with inflammation, and target those with already available drugs.
I mean, a couple of two examples to be very specific to your question. One is that as a company, we're developing sirukumab. We're testing sirukumab in phase 2. So this is a molecule which is in late stage for rheumatoid arthritis. You know, as part of the work we did with the MRC, we were able to identify. You know, part of it, there's a hypothesis, there's animal work, and there was some data that we were able to go back and reanalyze mood markers from RA trials and so on, and suggested that actually this was a good hypothesis to test. Is an anti-IL-6 monoclonal antibody, will that be effective in symptomatic depression? So that's one, you know, very simple example. The second one is, again for NEMA, I'll keep on referring to it because I think it's something that's going very well. The idea is, again, you know, in parallel work packages, can you identify those subjects which both from immunophenotyping, peripheral immunophenotyping and central immunophenotyping, can you identify the right subjects to do the trial in? I think the infliximab trial was very promising, but it was not, there was no patient selection. It was only post-hoc analysis suggested that if there was high levels of CRP, you'd see a benefit. And I think also to the point someone made this morning about who will pay for this, you know, to make the case for an expensive monoclonal antibody against a generic SSRI, you're really going to have to identify the right patients and show there's a real benefit over and above generic treatment.
That said, it's important to realize that there's still millions and millions of treatment-refractory suicidal depression patients out there who haven't responded to 8 trials of medication, even ECT. So it's not like there's not a large population.
I think, for us exactly, there's a very clear problem. There is a massive proportion of patients who don't really respond that well, and so there's a gap for better treatments for depression.
intervene
me to take that? Well, we're not as far as we'd like to be. I think there are plenty of papers, everything from looking at behavioral data to looking at early cognitive measures to looking at early subsyndromal behavior and symptoms. But at the same time, and there's certainly genetic approaches as well, I think it is the risk-benefit issue of, the issue of sensitivity and specificity. Everybody has bad days. Many people have bad weeks. How do you tell from those data who's really going to develop the severe refractory depression? And that the field hasn't quite connected yet. But I think if we can figure out as a field how to better take advantage of these already ongoing large-scale studies or large-scale scale collaborations, there may be opportunities for identifying biomarkers in a way that no one group is going to pay for.
I mean, in the UK, there's a lot of work done at King's and UCL and Cambridge, amongst others. There's members in the audience here, again, asking that question. Can you identify an adolescent who ultimately might become an adult, suffer of depression?
Any other questions from the audience before we... Yeah.
In terms of therapeutic modality, are we thinking about CNS penetrant drugs, or do you think it's peripheral inflammation is still...
I think there's evidence for both, and there's also really interesting preclinical data of replacement lymphocyte work and some of the same things we're doing in cancer now with sort of training of T and B cells. There's evidence of white cells that can actually integrate into the brain microglia space, and so I think all of the above is an opportunity. And Sean had a point.
I was just going to say, which makes a great point in terms of opening up those modalities beyond just a simple small molecule and thinking about how you can actually link the two together, I think is really important as well. So, yeah.
So, if one assumes that there's different kinds of depression, one thinks that some depression is associated with dementia, through possibly many different mechanisms, right, including co-location of genes, co-location of immune response, perhaps of neural death. Is there a type that you think of depression that's more specifically associated with an event in dementia? And then could that be used to help predict? So if you have this 30-year runway,
Yes.
it's going to be hard.
Exactly. So was at dinner last night, we were talking with Risa Sperling and others in this, where are there already ongoing longitudinal dementia trials where people were identified in early MCI or even pre-MCI that they didn't look at depression and and can we go back and do some of that as well? Almost certainly that is going to be the case. Also, almost certainly it's not going to rely on the current questionnaires we use. The Beck Depression Inventory and others, are you sad? Are you eating? Do you enjoy sex? Those sorts of things. Those are all important. They likely, I think the best metaphor is that we are with depression now where we were in the 1800s with the cough. You had these symptoms of depression. We had a cough then. You could do a cough suppressant, but we didn't know anything about the underlying heart failure, pneumonia, bacterial pneumonia, the variety, 20 different things that would cause it, and that's where we are now. Until we can have those biomarkers, I think we're not going to be able to have the depression subtypes overlying with the specific dementia risks.
I think it's fair to say the answer to the question is, we don't have the answer, but it's clearly a component that's critical if we're going to be successful. One of the studies that we're planning to look at now as well is as we're going into thinking about technology and how to detect these very small changes in cognitive function, to also ask questions in the other areas of the symptomatology particularly around depression, early child, all these components, because they're clearly going to be tied in, and ultimately then hopefully would give you some sort of readout that would be able to assess the patients that are most at risk. But it's clearly the key question if you're going to treat early in terms of being able to do that.
So, one thing that's important to remind ourselves of is that though talk therapy on the face of it looks simple and non-biological, there's now an enormous amount of data that the right kind of talk therapy, particularly the empirically validated ones, change the brain. They change the circuits, they change the synapses, and they can reduce inflammation. And so, I think as we continue to enhance our understanding of these approaches, I think there's a lot of room to grow there. And one thing we haven't talked about, but I thought I'd throw out there as well, is, there's the discussion of about Alzheimer's treatments that are in the non-amyloid, non-tau space, and enhancing neuroplasticity is clearly one of those. And places where we can identify molecular-based enhancers of cognitive plasticity will have extreme ramifications, both in cognitive disorders, but also depression and stress-related disorders.
I mean, to the point made earlier, I think about who and why and when, which patients, what subjects, and so on. I think that's a massive gap in the field, both for perhaps maybe more tractable in depression, but it's a massive gap. And I think we heard earlier in the last session about, well, if you are targeting inflammation or immunology in Alzheimer's disease, which subjects, what time during the disease course, and so on. And as far as I can see, there doesn't seem to be a very systematic approach to this research. It seems to be, there's a lot of meta-analysis, there's a lot of PET studies in subjects at a particular stage, but no real sort of longitudinal studies. And I think, to me, that's a real gap. The challenge is, I think, as Jacob Hooker said, that essentially the tools available for measuring microglial function in the brain just aren't good enough at the moment. You can maybe look at a trait effect, but not really a state effect. And I think that's the real challenge for us, I think.
So before I go back out to the audience, I just want to sort of switch gears back. And we mentioned at the very beginning, but low-hanging fruit. So what are the opportunities that we can do in the short term? I think we heard particularly loud and clear that there is no real long term unless there's a short term as well. And I think that's particularly true of any form of research in general. Any areas that we think are really areas certainly from the depression standpoint that we should invest in now that could tie into that patient population? What's the first area we want to go?
I mean, I think as I outlined, low-hanging fruit is a relative term, as you know, in the industry. But
how low does it have to hang?
Yeah. I think, there is, in depression itself, rather than depression as maybe associated with Alzheimer's disease, I think there are some very clear, we have some good molecules now to test the hypotheses. And I think, those are, I think, tractable experiments. We don't know the answers. We think there will be a positive, but we don't know the answers. I think it's harder in Alzheimer's disease. That stays in the obvious. But I think there are actually now tools. I think there's a willingness to look at the area of neuroimmunology, and there are tools to try and test the hypotheses. But I think it comes back to, again, it's a much harder question about who do you treat and when do you start treating them.
I think there's a number of places, and I think the low-hanging fruit are where there's already existing progress in one area, and we can just bring it over. And so I think that whether that's large-scale dementia trials, where we add more digital phenotyping and depression and psychiatric phenotyping, or whether that's large-scale psychiatric trials where we do the opposite with cognitive function and cognitive and peripheral biomarker measures. I think it's drugs in the anti-inflammatory space that are already FDA approved, already safe, and then identifying the subsets of patients and using the well-established animal models to really cross-transfer those two, as well as things like BDNF, esketamine, other things that are already being identified as very clearly involved in enhancing neuroplasticity that may be novel ways of trying dementia space as well.
What types of, going back to this idea of identifying these early populations, let's think about the tools that are going to be needed. What types of tools do we think we need in order to be able to sort of ask the right questions, particularly in these subsets of dementia patients that may have this sort of early symptoms that ultimately link into psychiatric endpoints? Do we have the tools? Do we need to develop better ones? How's the sensitivity measure?
I think it's digital phenotyping, it's biomarkers, both peripherally, and how can we maximize imaging biomarkers, particularly in the MRI space as opposed to dependent on PET per the prior conversations.
Yeah, I think, to my answer before, I think being able to show target engagement with a molecule, as you know, in this field, particularly for Alzheimer's, is absolute paramount, just because of the cost of a phase 2. So if you think you're affecting microglial function, how can you measure that in the CSF, in the CNS? And I think that, to me, is a real gap and a real need.
And I think one thing along that line that NIH has done a good job with, NIMH, with the RDoC initiative, is really saying they're taking a step back from clinical trials in psychiatric disorders, particularly stress, distress, depression, and stress, and requiring stage 1 target engagement before any other clinical trial can be done after that. So I mean, really, I think taking a piece from the playbook of neurology in that sense.
And I think ultimately that's going to obviously help then at least be able to define
and de-risk
the questions, right, as we go along as well. Time for a few more questions because we're actually going to run out of time in about 2 minutes. So we have a couple more questions.
CNS spends 30% of the time in a state called sleep. And I haven't heard anything that here so far which actually ties that state. I mean, we know it's involved in inflammation and in depression.
Yeah.
How do you think as a team
it's there's tremendous excitement there, I think, and the availability now of much better apps, wearables, and other things in the biometric space so that you don't have to go into an EEG lab, sleep lab to do it, where anybody at home, you can actually measure quite a bit of their sleep, as well as a lot more work in rodent models, in transgenic models where we can measure sleep EEG. So the data's just starting to come on. It's part and parcel and a really great biological readout as well.
I mean, I guess it's certainly an area we're interested in, and I guess just with the fundamental understanding of the importance of sleep, either for mood disorders or for Alzheimer's disease. But I think our understanding of the role is probably nascent. We do have some drug discovery programs in development that directly affect sleep, and the belief is that they will then have fundamental neurobiological effects. But I think it's a sort of an obvious. It's a fundamentally important. But I think the understanding, particularly for Alzheimer's disease, is still ongoing.
Time for one last question before we end.
Over here. Oh.
I have a question related to animal modeling of depression. As was said in the previous session about the limitations of animal models, and there is one field where there are real problems.
Yep.
Is this modeling? So at some point, some return to modeling will be required, even if you subphenotyping of patients.
Yeah.
What are your thoughts on this?
Yeah, so animal modeling of psychiatry has, I think part of the reason for the pullout of Big Pharma was that what animal models of depression did was allow us to reinvent more SSRIs, but that we were stuck there. And I think where we are now is a much better appreciation that we can't really create depression or PTSD or anxiety in a mouse that is, it may look face valid, but not really. But where we do have good modeling is of the specific circuits and the specific cell types. And so combining really the intermediate is the intermediate phenotypes in humans, the imaging phenotypes, the biophysical phenotypes. We model those. And then those connect to the humans. And I think great progress, especially with circuit tools like DREADS and optogenetics and other things, is making it much more possible.
So we've had our official indication that we need to stop. So I certainly wanted to thank...
quality control.
Yeah, it was. I want to thank the panel. Thank you for entertaining us as well with your questions as well. And hopefully this opened up some other aspects beyond sort of the components we've been discussing this morning.
Thanks so much.